All right, so we're gonna get started with our week 4 review session. I'm Tina.
I'm Melissa.
Yeah, so this is week 4, so that's gonna be like transcription factors. What's it called? The trip operon, RNA interference, things like that. Okay, so just a few announcements, like reminders. The problem set, assessment quiz, and discussion forums are due as usual Sunday at 11:59, and then we are going to have a exam review session on Tuesday. It's going to be 7 p.m. The room is still to be determined, so definitely Professor Francis on Monday, if she knows by then, she'll like announce where the review session is going to be. We'll send an email too. And then the exam itself is Thursday, September 30th, and all that information's on the Moodle page as well. And obviously, if you like are a select few who had different times makeups, that's like throughout the day. So all the information is on Moodle.
Okay. We are starting off with transcription factors. So we're asking, why do we have transcription factors? And we have transcription factors because RNA polymerase, which is used for transcription, cannot bind to the DNA on its own. It needs something to help it attach to the DNA. And so here we have a picture of RNA polymerase with a sigma factor, which is helping it to perform transcription in prokaryotes. And then the RNA polymerase will dissociate from transcription factors once the transcription has been initiated. Okay, so as I said, transcription factors control the RNA polymerase binding to promoters in the genes and DNA. There are two types of transcription factors that can be found in cells. The first is a general transcription factor. These can be used at many different promoters. They're not specific to any type of gene in the DNA. Some examples are sigma factors, which are found in prokaryotes, and basal transcription factors, which are found in eukaryotes. Something about sigma factors is that they can be used at many different promoters, but they can also act as a regulatory transcription factor. So we're going to get into that in a second. And then regulatory transcription factors are the ones that control specific genes at specific promoters. And so there are two types of regulatory transcription factors. There's positive and negative. So positive basically enhances transcription when it's bound to DNA. It allows the RNA polymerase to bind and transcribe. And then negative does the opposite. It suppresses transcription when it's bound to DNA, so it doesn't let the DNA polymerase bind so that it can't transcribe the gene.
And just really quick, I forgot to say it at the beginning. We have like question slides built into the slides. But if you have questions like while we're talking, definitely stop us whenever.
Yeah. So sigma 70 is a type of sigma factor that is, I believe, in prokaryotes as well as eukaryotes. So this is necessary for the transcription in prokaryotes. So the sigma factor has to bind to the RNA polymerase before it can bind to the promoter. And in eukaryotes, it does not work like this. It binds this way because it helps the polymerase bind to the gene in a sequence-specific way. So after polymerase binds, the sigma will leave after transcription is initiated because its job is done. And so it'll dissociate and go find another RNA polymerase that needs to be bound. And it unwinds, it binds at the -30 box and the -10 box in the promoter. And so the initial unwinding will take place at the 10 box because there's more AT base pairs there. And as we know, AT base pairs have 2 bonds while GC base pairs have 3. So it's more energy efficient for them to begin unwinding where they only have to undo 2 base pairs at a time. Sigma 70 is sequence specific. It has a consensus sequence, but not all promoters have the sequence. So depending on the affinity of a promoter to sigma 70, that'll alter how often that promoter is transcription begins at that promoter.
And I just want to add, like, to clarify a little bit. So sigmas are general transcription factors. And then the definition of general transcription factors is that they're not sequence specific. But then you might think it's like contradictory because we're like, oh, they have to be at -35 and -10, which makes it sequence specific. So the only difference is that it is general because it can be at a lot of different promoters in the prokaryote. The -35 and the -10 is, like Melissa said, the consensus sequence. So it doesn't have to be exactly these two sequences every time, as long as it's close enough, so to say. It can still help RNA polymerase bind to the DNA strand. Does that make sense? So even though it's a general transcription factor, that means it can bind everywhere, but it does have a preferable sequence that it will bind to with the highest affinity. Does that make sense? All right. Is this me? Yeah. Okay. So going on to gene regulation. So anything about transcription factors that you guys had questions on before we move on to a different topic a little bit?
Are they all proteins?
Are transcription factors proteins?
Yes.
So transcription factors are proteins, yeah. So gene regulation, and we're going to be focusing on prokaryotes in particular. So in this case, we're talking about the trip operon in this class. So an operon is a set of genes transcribed under the control of a single promoter. And then just the terminology, polycistronic mRNA. So that means it's a single mRNA that can code for multiple proteins. So for example, this is like one single mRNA, but it has a lot of different genes encoded within it. So a single mRNA can code for many different proteins. In this case, it can code for proteins A, B, and C. And then the transcription, oh, sorry.
Does A, B, C have like their own starting and termination amino acids, or is it like, do they just start coding or like translating it and just never stop until the end?
Okay, yeah, so it would transcribe the whole thing because it's one operon. So once it starts transcription at the promoter, it's going to transcribe A, B, and C. Like it won't just do A and then stop and then B and then stop. It wouldn't do sectionally. It would be all of it. Yeah, so then transcription of operons are controlled by different transcription factors. You can have activators, which increase transcription, and then you can have repressors, which will prevent transcription. The trip operon that we've been learning this week, that is an example of a repressor, which we'll go into more detail. But activators and repressors can be turned on or off. So depending on whether they're activators or repressors, they can be like a negative feedback loop or a positive feedback loop. So I just want to make sure. Yeah. Okay, so each operon contains regulatory DNA sequences that act as binding sites for regulatory proteins. So basically that just means within the operator, there are promoters and there are operators. So the promoter is where the RNA polymerase binds to, whereas the operator is where a repressor will bind to. In our case, it's a repressor. It could also be activators, depending on what operon you're talking about. So for example, in this case, operons can be inducible or repressible. Inducible means that the operon is usually off, but it can be turned off. Sorry, it can be turned on. And then repressible is it's usually on, but then the opposite, it can be turned off. So the trip operon is repressible because usually it is on, but when the repressor is bound and there's a lot of trip around in the cell, it will bind to the operator and then that turns the operon off. Yes.
We didn't learn that in class. Electric. We have to know it.
For which part?
The inducible and the end part.
Oh yeah, so inducible, that's just like the opposite of repressible. I don't think you learned it specifically because we're learning about the trip operon, which is repressible. I think if you learned about like the lac operon, she talked a little bit about that from like your freshman bio class, that would be inducible. Yeah, but you don't have to know too much about inducible for this class. Okay, so specifically focusing on the TRP operon, TRP stands for tryptophan, which is a amino acid. I think one of your questions on the problem set this week was asking if tryptophan itself is a repressor or not, and the answer would be it's not a repressor. It can be a co-repressor in this case, but tryptophan itself is just an amino acid. It doesn't really do anything on its own other than act as an amino acid to add onto a polypeptide chain. So there are different scenarios within the TRP operon. There should be 3 scenarios here, but I just wrote 2 for now. But you can have low tryptophan levels, and that means your bacteria does not have access to tryptophan. So that means if there's no tryptophan around, they have to definitely make more tryptophan. And so the operon will be active, it'll be on, so that the prokaryote can transcribe and translate to make more tryptophan. On the reverse hand, if you have too much tryptophan, if you have it in high levels, then that means there's plenty of tryptophan for the bacteria to have access to. So they don't need to have that operon on because they don't need to produce any more tryptophan. So that operon is off in this case. Oh, and then just a little question. Is the tryptophan repressor protein a positive or negative regulator? So not tryptophan itself, but the tryptophan repressor protein. Does anyone know if it's positive or negative? Yeah, nice job. Negative, cool. Okay, so this is just a little diagram, like a closer diagram of the tryptophan operon. So you have the repressor. So this is the DNA strand, and then this is your repressor, because in order to get the repressor, it has to code for it. So it'll code for the repressor, and when there's no tryptophan around in the cell, there's nothing to bind to the repressor. So that means this is off. And if that's off, there's nothing to bind to the operator. So the entire operon is on. So that sounds confusing, but if this is inactive, then that means it can't bind, so it's not repressing it. So the operon is on. Does that make sense? Okay. So then when there is tryptophan around, it will bind into the little binding site, and then that means the repressor is active. And so since it's active, now it can bind to the operator. And then when the repressor is bound to the operator, it's kind of like blocking the promoter site. And that's what's making it a repressor, because if it's kind of blocking off the promoter, the RNA polymerase cannot come in. There's not enough space for the RNA polymerase to bind to. And then that's what makes the entire operon off. This is the leader sequence that we'll go into more detail about, but there's the leader sequence is where like attenuation occurs or early termination transcription. And then these are all of your different tryptophan genes that come from the same single mRNA strand. Ooh, it feels so nice. Oh no, sorry. So going into the different scenarios, so there was like low tryptophan, high tryptophan, or medium tryptophan. So we're going to go into each one in detail. So the first scenario is when you have high levels of tryptophan. So what do you think will happen? Will the operon be on or off? Will transcription occur? Will tryptophan protein be made? The operon will be off and transcription will cannot occur. Yeah. Yep. So that means the tryptophan is bound to the repressor because there's enough for the repressor to be bound, like it was that pie shape that I had before. So tryptophan is bound to the repressor. That means the repressor, which is shown in like that little pink color, can bind to the operator. And then if the repressor is bound to the operator, it makes it not enough space for the RNA polymerase to come in to bind to the promoter. And if the RNA polymerase cannot bind to the promoter, that means there's nothing to initiate transcription. So you get no transcription. Okay. So before we go into the next scenario, which is where attenuation can occur, just a little review about what attenuation is. So attenuation is the early termination of transcription. So it just means that transcription ends early. Your attenuation sequence comes before the DNA coding for the TRP proteins. So sometimes in your like problem sets, I think she calls them leader sequences. So the leader sequence is the attenuation sequence, and it comes right before all of the TRP DNA code genes. And then there's 4 regions for the TRP operons leader sequence that can base pair with each other and complement each other to form these like hairpin loops. Sometimes they're also called stem loops. So 1 and 2 are complementary, so they can pair together. 2 and 3 are complementary, those can pair together. And then 3 and 4 are complementary, so those can pair together. You won't get like 1 and 4 or 1 and 3 because they're just not complementary.
And these hairpins occur in mRNA?
Yep, so this is the mRNA strand. Yes. So depending on what regions pair together, it causes different effects. So if 1 and 2 come together, it causes a pause in transcription. If 2 and 3 pair together, that means transcription will continue. And if transcription continues, that means translation will happen. And then if 3 and 4 come together, that is when attenuation or early termination of transcription will occur. And then this only happens in prokaryotes, I want to emphasize. So eukaryotes don't do this. The only reason that prokaryotes can attenuate is because they transcribe and translate at the same time. So like as the RNA polymerase is transcribing this mRNA, like the, what's it called? The
Ribosome.
Oh yes, the ribosome can come in behind it and like latch on to the strand that is coming out behind the
Polymerase.
the polymerase. Sorry guys, the strand that's coming out of the polymerase, ribosome can come in to bind that. So it's like transcribing and translating at the same time. All right, so that leads to the second scenario where you have only low levels of tryptophan. So there's not enough for the cell. So what do you think will happen like operon wise? Will transcription occur? Yeah, all right, cool. So yeah, so tryptophan will not be bound to the repressor because there's not much of it around. So if it's not around, it can't bind. That means the repressor is inactive, which makes the operon active. And so RNA polymerase can thus bind to the promoter because there's no repressor around to kind of like knock it off, and it'll start transcribing. And there will be no attenuation in this case because it wants to make the full transcription of the mRNA in order to get a higher level of tryptophan in the cell. So this shows a better illustration of what's happening. I kind of broke it up into like 3 segments. So first, what happens is your 1 and 2 regions will come together. So the RNA polymerase, if you can picture it starting off in, oh, can you see my mouse?
Yeah.
Oh, awesome. So if RNA polymerase, if you can picture it starting off over here in the promoter region, it was going across transcribing. So it transcribed 1 and 2 already. And that means 1 and 2, since they're complementary, they can come together first. And that causes a pause. And the pause is important because it lets the ribosome catch up to the RNA polymerase. So that's why RNA polymerase and the ribosome are co-reliant on each other. Yes. I don't know specifically. We've learned that because I've been told at least that ribosome and polymerase, they're codependent on each other. And so if the RNA polymerase is going too fast, then ribosome can't catch up, and that's like detrimental for the cell. specifically why it causes it to pause, I think that's a signal that the cell has come to adapt to. I can ask that though, and then I can get back to you.
I think I remember this was back when I took the class back like 2 years ago. So this might be wrong, but I think I remember part of it was like the ribosome could not physically attach to where there's double bonding. So it has to wait for it to bond, and then it has to wait for the bond to come undone. And then the ribosome can keep moving. So if the hairpin is just momentary, so the ribosome has to wait till the hairpin is come undone.
But the RNA polymerase is already ahead.
Yeah, I think he was asking why does
Oh.
1 and 2 cause RNA polymerase to pause? Like what's stopping it from
Oh, then I
continuing on?
don't, sorry.
Yeah, I can ask that for you, and then Laura can probably address it in class if I let her know.
That's fine. I just was confused before because I thought the RNA polymerase was find it.
Oh, no, it's in front of it. Yeah, so we'll see here that, so because it's paused, the only way that it can keep on going is the ribosome is basically gonna like knock into the hairpin stem loop. Professor Francis describes it as like a car crash, like it knocks into the hairpin loops and like knocks it flat again. So that comes to the second scenario. So it's gonna keep on going, it knocks it flat. So now region 1 is flat again for ribosome to keep on going. But this is the trip codon. So the trip codons are in region 1. And because there are very low levels of tryptophan tRNA around, it's going to be stuck there for a while because there's no tryptophan tRNA around to add to the polypeptide chain. Does that make sense as to why it's stuck there?
You want to explain that?
Yeah, that's fine. So ribosome is what creates the amino acid chain, right? So once it gets to the tryptophan codon, that means it needs tryptophan to add to the polypeptide chain. But since there's not enough of it around, it's going to be really slow to add tryptophan to the chain, because the addition of amino acids is all random. Like it's not like they're calling it in. You have to wait for it to kind of like float into the ribosome area. Yes.
So RNA polymerase can't move until it gets stuck there.
Oh, are you asking like when RNA polymerase is going to start moving again?
Yeah.
So RNA polymerase is going to start moving again once ribosome knocks down 1 and 2.
But if it's stuck there forever, or is it usually?
No, it's not stuck there forever. Yeah, so it's only stuck momentarily. It's stuck long enough for RNA polymerase to keep on transcribing.
But how can RNA polymerase keep on transcribing? Because before it couldn't because the ribosome was close. Now it's close enough while it's stuck.
So it was paused because 1 and 2 were complementary together. So 1 and 2 is the signal to cause the pause. So now that 1 and 2 are not together and 2 and 3 are together, that means ribosome can the RNA polymerase can continue on. Yes.
So the ribosome, so when 1 and 2 pair together,
Mhm.
RNA polymerase pauses, then RNA polymerase doesn't continue until the ribosome like crashes into the
Yeah,
protein
until
and
1 and
knocks it,
2 are not
and then
connected
they both
anymore.
continue moving
Exactly.
until the ribosome gets stuck on the trip codons.
Yes.
Okay. But then RNA polymerase is still moving.
RNA polymerase is still moving.
Either
The only
one.
time RNA polymerase pauses is when 1 and 2 are together.
Okay.
But
In the back and then.
if the ribosome is stuck, then 1 and 2 can't come apart. So doesn't that mean RNA polymerase?
Oh, okay. So this is 1. And then this is 2 and 3. So in the first picture, 1 and 2 are together. So then ribosome comes over, knocks it down, and now 2 and 3 are like flat
the
again.
stop codon is in 1.
Yes, the pause codon. It's not a stop codon. It's the trip
Oh.
codons that's causing it to pause and get stuck. Does that make sense again? Yeah. Did you have a question?
That was my question.
Okay.
That was region 1.
Awesome. Anything else? Okay. Yes.
What happens? Because in my notes, it said ribosome's gonna slide to position region 2. And disrupt the interaction between 2 and 3? How does it do that to stuck?
It disrupts 2 and
3.
3.
Conforming, allowing 3, 4 hairpin to form.
I think it's because it's not stuck forever. It's just takes a while. It takes a pause
Yeah.
because there's not a lot of trip in the cell, but
Mhm.
eventually the trip will
Yeah.
attach and it will be able to keep on moving.
Yeah. Sorry. So maybe stuck isn't the right word. It's just like a longer pause, waiting for whatever tryptophan there is to eventually make its way to the ribosome, and then it can keep going.
Okay.
But it's just paused long enough. So the important part for why it's paused there is that it's paused long enough for RNA polymerase to continue transcribing.
Mhm.
So then that'll transcribe region 4 over here. And so because 2 and 3 were complementary and 2 and 3 were paired together, even though RNA polymerase has now transcribed region 4, attenuation will not occur, because the only way attenuation can occur is if 3 and 4 are together, but 3 is preoccupied with 2. So that's why attenuation will not occur. Does that make sense? Okay, so eventually ribosome will become unstuck and like unpaused because tryptophan will eventually make its way to it, and then it'll keep going, knock this down, and then just continue on normally. Does that make sense?
Is this just for the low levels?
Mhm. Yes, this is only in the case of low levels where attenuation will not occur. Yes.
So is it like that same thing where then when it gets enough of the trip codons, it like leaves again, then it has like another crash and it unbinds 2 and 3, and then it's just fine, it can be normal, except like how do they unbind it?
How does 2 and 3 unbind? Yep, so it's the same thing. So once there's like some tryptophan, eventually it makes it to the ribosome. The ribosome can continue transcribing. It'll knock into 2 and 3 and like flatten it out. And then transcription occurs normally.
And then at that point, 3's not gonna.
Yep, because at that point, it's just like the timing, because the timing is hard to show through pictures. So it's just timing wise. At that point, 3 and 4 won't be able to bind together because most likely at that point, the ribosome will have gotten to like 3 or something.
Okay.
Yeah. So it's just hard to picture exactly because it's like still pictures, but this is like a life and moving interaction. All right. Does that clarify a little bit more for everybody? All right. And then if you guys have more questions later on, by all means. All right. So then this is the last scenario for the trip operon. So you could, she calls it medium in the problem set, but essentially that just means like maybe you started off with low levels and then the cell realized that it actually has enough. So in order to not waste its energy, it's going to want to cause attenuation. Oops, I answered the question, but attenuation will occur. So like transcription will start to occur, but eventually it'll stop because attenuation will happen. So initially, tryptophan is not bound to the repressor. So that means you have an inactive repressor. And so the repressor is not bound to the operator. So transcription will start to occur because RNA polymerase can bind. But then the cell realizes that there actually is enough tRNA around. And in order to not waste its energy, attenuation will occur. So you'll have a short mRNA transcript in the results. So this is the mechanism for low to high levels or medium levels. So in the beginning, it's the same thing. You have RNA polymerase starting over here. It's moving along. It makes 1 and 2. 1 and 2 come together, and that's like the pause signal in order for ribosome to catch up. And this is where it's a little bit different. So ribosome is continuing on, and it won't be paused long in the region 1 where the trip codons were, because whereas in the other scenario, there was not enough tryptophan around for it to move quickly. Here, there is enough tryptophan around. So tryptophan goes through and gets added to the amino acid sequence quickly. So ribosome moves over region 1 quickly until it reaches the stop codon right before region 2, or kind of like blocking region 2. And so since ribosome is kind of stuck around the region 2 area, that means 2 cannot bind to region 3 because the ribosome is there. So it's like a blocker from those 2 pairing together. And on the other hand, RNA polymerase, there's nothing stopping that from moving on. So it continues on to transcribe region 4. And since region 3 could not bind to region 2 because of the ribosome, that leaves 3 open to bind with 4. So that's what happens here. 3 binds with 4, and that is the attenuation signal. So essentially what happens is this is the binding site in the RNA polymerase. So when 3 snaps together with 4, it basically like yanks it out of the binding site in the RNA polymerase. And that's what causes attenuation to happen, because if it's not in the RNA polymerase anymore, then it can't keep transcribing. Does that make sense? You have a question?
Why doesn't the ribosome get stuck on the stop codon one in the low, like example? Or
Why
does
does it not? Oh, so like, why doesn't it get stuck here?
Yeah.
So it will still stop, but by that point, because the 3 is blocked from binding to 4, it still won't matter, because eventually it'll keep moving after it realizes that the stop was not useful, or like that it didn't need to stop. It'll keep moving, and then just timing wise again, the 3 won't be able to pair with 4.
So it's just a temporary stop in the flow.
Exactly.
It doesn't
It's
affect it.
yeah. So it picture wise, it says like stuck or stop, but really it's just a longer amount of time for the cell to figure out whether or not it actually needs to keep going or to stop early. Okay, so that's just like a simplified summary, like a different way of looking at it in a more simplistic way. And then the slides are going to be shared with everyone too. So if you ever want to go back to this slide, that might help you out. Does anyone want me to stop on this slide a little longer, or can I keep going? You want to stop here?
For situation 3,
Mhm.
Does it still form short protein, or is it just?
Nope, because it never, so remember that the leader sequence, so these like regions 1, 2, 3, and 4, they are before the genes that code for the proteins. So if 3 and 4 come together, it yanks it out of the RNA polymerase before it can start to code for the proteins.
Okay.
Yes.
In the problem set, like it asks about like a situation concentration, is that like situation?
So situation 3 means medium. Yeah. So low to high is the same thing as medium.
And in the medium one, is there a repressor also binding to the operator sometimes?
Not initially, because for the medium scenario, transcription must have started. It's just that there's still enough around for it to end early. It's kind of like the cell's like in conflict with itself, where it thinks it needs it, but then it realizes that it messed up and it doesn't actually need it. So it's like, oh no, let me yank it out. Yeah. Does that make sense? All right. Okay, so that's just like a question slide.
Okay, now we're moving on to non-coding RNA, which is essentially just any RNA in the cells that is just not mRNA, so it's not coding for a gene. So he said, non-coding RNA is RNA molecules that do not code for proteins, so don't undergo translation into a polypeptide chain. Instead, they have a lot of functional properties in our cells. They can work as enzymes, which are known as ribozymes, and they catalyze chemical reactions. Or another example is they can help with regulating gene expression, like helping with translation or the splicing of DNA into introns and exons. Okay, so there are 2 main types of non-coding RNA that you have to know for this class: ribosomal RNA, which is rRNA, and transfer RNA, which is tRNA. A ribosome is made of two protein subunits and then also rRNA. And the rRNA is the part of where translation occurs. So it acts as a catalyst for the polypeptide bonds that are formed between each of the amino acids in the polypeptide chain. And so this is called a ribozyme, like a combination of the words ribosome and enzyme. That's a good way to remember it. And then tRNA is transfer RNA, which brings the amino acids from throughout the cell to the ribosome, so that they can be synthesized onto the polypeptide chain. And how this works is that the tRNA, I'll show you that later. Sorry. The tRNA here has three bases that will pair with the codons for each amino acid. So like these will have opposite base pairing for each specific amino acid. And so that's how that gets added. And then it releases the amino acid once it's in the ribosome, and then it will be attached to the polypeptide chain. Sorry. Okay, so there are other notable types of RNAs that we just want to touch upon that aren't as important. There's small nuclear RNAs, which are the catalytic part of spliceosomes. These splice the RNA into introns and exons. Then there's long non-coding RNA, which is LNC RNA. This is 1700 base pairs long about, and that's mainly involved in X chromosome inactivation with the genes exist and T6, which are on the X chromosome. There's also CRISPR RNA, which is a commonly used bacterial defense mechanism. This is where the guide strand is used to target an invading virus of DNA, and then it cleaves the DNA of the viruses. This mechanism is being used a lot in research to knock out or insert certain genes in eukaryotic cells. SRP is a ribonucleoprotein used in protein translocation to the ER. And then RNases are RNA nucleases, which cut RNA in nucleoproteins, in which the RNA portion is the catalytic part. So the RNA itself is what is doing the cutting.
So you don't have to know like specifically each one.
Yeah.
I think she just mentioned them in her lectures, so we thought it'd be useful to just kind of put them up here for future purposes if you want them.
Mm-hmm. And so now,
Please. Oh,
okay, now we're going into RNAi, which is the process of RNA interference.
It's just me being corny. Sorry.
So there are different types of RNA control, and this one is post-transcriptional, so it occurs after the RNA has been transcribed from the DNA, but before it becomes a polypeptide. This is found across eukaryotic cells. And so this works for suppressing gene expression, and this also can be useful in research for knocking out genes. So to see like what the gene's function is if we like turn the gene off, and seeing what stops occurring. And so we're going to focus on the RISC, which is the RNA-induced silencing complex. This is a protein complex that uses miRNA and siRNA as guide strands. I'll get more into that in a bit. The miRNA is used as a guide strand for endogenous gene suppression, so suppressing genes that are within the cell system. And then siRNA is exogenous gene expression. So this will suppress genes from like a bacteria or a virus, so those can't be reproduced in your own cells. So the RISC is involved, like I said, with the process of targeting mRNA that codes for proteins in the cell and altering it so that it cannot be translated into a protein or used in the cell. The general mechanism of the RISC is that it unwinds and it will bind to a guiding strand of DNA. So the guiding strands of DNA are initially double-stranded, and it will unwind, or not DNA, the guiding strands of RNA are double-stranded. So then the RISC will unwind that and bind it to it. And then this strand will allow the RISC to target mRNAs that are complementary base paired to either the miRNA or the siRNA. Does that make sense? Yes.
So miRNA is for stuff that's in our cells already?
Yes. So yeah, microRNA is endogenous, which is like our own genes. So maybe if we're making too much of something, it will stop so that the cell doesn't waste energy on continuing to produce that or materials on that. And then siRNA would be like bacteria or virus that is harmful to our cells, RNA from those.
The terms endogenous and exogenous, it can also clue you into the pathway, like the difference between the two pathways. So because mRNA is endogenous, it starts off already inside your cells, whereas siRNA has to come from some outside source, like the viruses or other bacterias.
Yeah. Okay. So any questions on that
So far.
so far? Okay, we're gonna get into the more specific microRNA. So microRNA is the one that is endogenous. So for things within your own cells. Oh, I meant to say earlier also, other organisms have these types of processes, but we're going to be focusing just on human cells. So like I said, this process is post-transcriptional regulation, and mRNA is one of the guide strands for the risk to target and base pair with mRNA. This base pairing allows it to regulate the stability and presence of the mRNA. And so keep in mind that these are double-stranded. They are about 20 to 25 base pairs. They're pretty small. And yeah, they're endogenously expressed. So they're within the genome and then they're expressed in the nucleus. And they're not very prevalent in humans. So they're thought to regulate about less than 60% of humans' protein coding genes, not as much as siRNA is used. So this is the pathway. So we're looking at step 1 here, where RNA polymerase II will transcribe the initial transcript. So this is the pri-mRNA, and this is not yet pre-mRNA. So then in step 2, the first cuts are made by drosha. Drosha is an RNA nuclease. So a nuclease is an enzyme that can cut nucleotides. So it's cut into pre-mRNAs. Then the pre-mRNA is exported from the nucleus to the cytosol, and it's using export N, which we discussed last week in the nuclear export sequence. And then, okay. So then in the third step, sorry, another RNA nuclease called dicer will cut the pre-miRNA into just miRNA duplexes. And so these are now about the 20 to 25 base pair long, and they are ready to be used by the RISC. So this is what is picked up by the RISC and unwound and used as the guide strand. So here is the, here is the duplex unwinding. This is RISC unwinding, and then coming into RISC, and then the next step, here it is as a guide strand. This red one is the guide strand. So it will complementary base pair with the target mRNA, and this green strand here is the target mRNA. So the end result will typically be suppression of translation, which will either occur by, the mRNA is either destroyed completely, or it will be sequestered into P bodies, where it can be here for decay, or it'll be stored there if it is needed later in the cell's lifetime. Yeah. And the binding here is imperfect. When binding with the miRNA and the mRNA is imperfect, that's when it goes to the P bodies. But if it is perfect, then it'll just be destroyed. Does that make sense?
Yeah. All right. So small interfering RNA, which is the other, sorry, what's it called? Other guide
RNAi.
strand. Yeah, sorry, our other guide strand for RISC. So it is also a post-transcriptional regulator. So after RNA is made already, it's a guide strand for RISC, and it also regulates mRNA stability. So these are all similarities between the two. This is also a similarity. They are also very small, about like 20 to 25 base pairs, also double-stranded. The difference here, though, is that they are exogenous, like we were saying. So they are brought from somewhere outside of the cell, usually viral. That's one of the way that our cells are able to like defeat the viruses by getting rid of these viral RNA strands. Yep, so they can protect organisms against foreign invaders through the suppression of these viral infections. And they usually are perfect pairing in humans.
How can it protect the organism if it's just coming into the cell to destroy the mRNA?
So siRNA, so because it comes from outside, you want to have some way to get rid of those RNA strands, right? So by using siRNA, it can be complementary to those
So
viral.
siRNA doesn't come, wait, it comes into the cell?
Yep, siRNA is also comes into the cell. It's from somewhere outside.
Do you mind explaining again how it protects the cell? I'm not using something.
No, that's okay. So it protects the outside against outside invaders because so if the siRNA is coming from outside of the cell, that itself, even if it's viral, it gets cut up into like the small pieces again. So it's not like a full viral cell, but it can act as a defense mechanism for if a full viral strand comes in, and then it now has these siRNA strands, and then it has something to base pair with.
Like a memory in the cell.
Yeah, so something like a preventative measure for eventually, yeah. Okay, so the specific mechanism of the pathway, so like I said, it comes from outside the cell, but then the pathway itself starts in the cytoplasm, whereas miRNA started in the nucleus. So because it starts in the cytoplasm, the double-stranded or single-stranded RNA is cleaved by dicer to become the little smaller 20 to 25 base pair duplexes. RISC comes in to unwind those duplexes so that they can become single-stranded and become a guide strand for the RISC. And that is helped through the help of Argonaut, because Argonaut is the enzymatic portion of RISC. So Argonaut is doing the unwinding. So now you have the RISC siRNA complex, and this complex can now base pair with mRNAs that the siRNA is compatible with or complementary with. And so that is why it can be an exact base pairing match. So whatever complements siRNA, it exactly pairs with the mRNA, and then RISC can go in with its Argonaut 2 subunit, which is also the enzymatic portion. The Argonaut 2 is the enzymatic portion that actually does the cleaving. And so once you cleave mRNA, the mRNA is degraded or silenced because it just like chopped it up into pieces. So it's not useful anymore. Does that make sense? Okay. So this is, oh yeah.
I just have a question. What is dsRNA and SH?
Oh, sorry. DS stands for double-stranded RNA, and then SH is singles. Oh, no, it should not be single. Wait, what is SH? Single hairpin.
Oh,
It is
short
it
hairpin.
is
Short
short hairpin.
hairpin.
Okay, that makes sense because it's hairpin loop. Sorry, I should know that. But yeah, essentially it just means that it has to have like, it has to look at least, so this is a double strand, but this is technically single. It's just made
Folded on.
into a double, yeah. Because it has to have two in order for Argonaut to like unwind it. Yeah. Oh, and then this would help with your problem set too. So one siRNA is very versatile. Basically, you don't need a lot of siRNAs in order to cleave a lot of mRNAs. So because it's an exact pair, siRNA, it's not like energy where it kind of gets like used up. It's constantly there, or it can be for as long as the cell needs it to be. So RISC complex can continue to use that same siRNA to go and silence however much mRNAs that it wants to, or that it needs to.
And mRNA not do that?
mRNA can also, you don't need a lot to have like a big outcome, but just because mRNA is not an exact base pairing match, it can't do or be as efficient as siRNA. So that's why like humans don't really use miRNA as much, I think, right?
Is Argonaut still a part of the mRNA?
Argonaut is the enzymatic portion of RISC. So this is, again, another, I like to have like little like summary slides. So it kind of tells you the difference between miRNA and siRNA. And this shows the two pathways side by side. So this is miRNA, where it starts in the nucleus. And then I believe the right side is miRNA. And then the left side is siRNA. So yeah, so it's like a summary of the differences and the pathways.
So after like RISC and Argo and all these things like bind, and once it's degraded, does it like unbind and just do other things, or is that like part of it? And then it's degraded too.
Sorry, one more time. What gets
So
part of
you
it?
know when like you have the pre-mRNA and all that kind of stuff, and then you have the RISC and the Argo or whatever. So that binds to it.
Mhm.
When you're degrading the mRNA and all those things, are those degraded too, or do they unbind and then keep working?
Okay, so correct me if I'm wrong too, Melissa, but RISC and Argonaut are, it's their protein complex that's like always around in the cell for when it needs to be used. So they're never gonna get degraded unless like the whole cell is getting degraded. So they're just kind of like floating around. And then the strands, I think they're part of the complex for as long as they need to be. And that's why you can use them a lot of times, because it can be like reused for many mRNA strands. Yeah. So unless it needs to be, I don't think they get degraded.
I thought you said earlier exogenous means that it's from outside the cell. Or does it mean outside the
Outside of the cell, but we don't show it, the pictures don't show it coming from outside the cell. It's just showing that since it's coming from outside the cell, it doesn't start in the nucleus.
But in parentheses, it says like next to exogenous. Or like is that the definition?
The process starts in the cytoplasm. So it's coming from outside the cell, but it's like everything that's going to be happening to the siRNA strands starts in the cytoplasm. Yeah. Any other questions? Does anyone want me to stay here a little bit longer to like take it in? So that is the end of the review itself. So I'm going to stop the recording.